Thanks everyone for being here, and I'm pleased to take you on what I'm calling a walk through a random forest. And right off the bat, I want to let you know that this is kind of a lecture geared towards the quantitative noob, the student, or God forbid, the senior scientist not familiar with using machine learning. In that sense, it harkens back to kind of those for dummy or complete idiots guides. To sort of pull out of that nosedive of self-flagellation, I like to think about it as the prologue to a book entitled The Reasonably Bright Ecologist's Guide to Using Machine Learning to Answer the Questions You Really Care About. And so what I'm thinking of is somebody who's not necessarily yet super interested in the pedagogy of random forests, but who wants to pull it out of the box, kind of brush off the packing peanuts and start using these machine learning algorithms. And I've included in here kind of a poetic disclaimer by Alexander Pope, who says, "A little learning is a dangerous thing." And he goes on that, but you kind of get the gist. And that's that what I'm telling you right now isn't going to be enough to get sort of that deep drink of random forest to take you to those new distant scenes of endless science. I'm leaving you with just enough to leave you drunk and dangerous on knowledge, and then the rest is kind of up to you. So this is geared towards the newcomer to random forests. And I'm going to go ahead and do it sort of semi-autobiographically. So I'm going to say the first thing you need to do if you don't have enough already is get a problem. And so in my case, I walked in one day and my postdoctoral advisor said, "How would you like to map the world's symbioses?" And I said, "Okay." And I'm going to make a major simplification here because the world forests have, over 3 trillion trees with a T. They cover about a third of the world's surface. There's thousands of species and thousands of trait combinations. It's a massive sort of hyperdimensional problem. But the good news is that we can assign almost all, more than 99% of tree individuals to one of two symbiotic groups. So these are mycorrhizal fungi that belong to different functional groups. And these groups differ in how they cycle nutrients and carbon. And these differences can scale up to the whole forest ecosystem process. So there's a difference between how a forest dominated by one group cycles nutrients and a forest dominated by another group. So that if we had a global quantitative map of the distribution of these two symbioses, we would kind of have a map of ecosystem function. And that's the problem. That's what we wanted to do. And so the first thing we did is we wanted to look at the state of the field. And when we did that, we cracked open some biogeography textbooks. And we found these hand-drawn maps, which look kind of like biome distribution maps. And they have the presumed dominant mycorrhizas of different forests. And it's really based on kind of guesswork and some untested hypotheses. To me, I was kind of elated when I saw this was the state of the field because it all seemed kind of as speculative and pre-scientific as the idea that Helios marched across the sky in a golden chariot dragging the sun in his wake. So I thought we can do better. We can make a quantitative map as long as we had a ton of data. And so that's step number 2. We needed a bunch of data. And this is where we relied on GFBI. That's the Global Forest Biodiversity Initiative. And it's got 1.1 million forest census plots. And that means there's georeferenced trees, 31 million tree stems, where we have the size and identity of the tree, and they're spread throughout every forested biome on the planet. And it's a huge amount of data to have at your fingertips. And what we did is we used these published rosters of which tree species belong to which mycorrhizal group. And so we simplified this massive data set into, for each forest, the proportion of trees that belong to either ectomycorrhiza or arbuscular mycorrhizal symbiotic groups. And when we did that and aggregated to a grid, we got sort of the map that we hope to explain. So here we have our grid, and it's colored according to the relative abundance of 3 different symbiotic groups, the 2 major ones being the arbuscular and ectomycorrhizas. What we really wanted to do was build a predictive model that explained why we got this map, why the distributions are the way they are. And we wanted it to be stable and predictive enough that we could extrapolate symbiotic states throughout the entire world forest system. And I just wanted to mention as I'm going through this that I think that these kind of projects are actually a good way to cut your teeth on random forest problems. And that if you wanted to, you could apply to GFBI to use their data set and then link it up with other plant traits, for instance, through the tri-plant trait database. And you can build mean trait value maps over the globe and try to predict them with these machine learning algorithms. And I think that's a good way to, a good entry to random forests. So our third thing is we needed training data, right? Because there's a lot of different potential drivers of symbiotic state, an embarrassment of riches, and I didn't know where to start. And so there's also a lot of freely available global rasters of different environmental predictors that could drive symbiotic state. So you download those rasters and you extrapolate the data across, the same areas where you have forests, and you have a huge data frame. And then you get to the fourth part, build a model. And this is where, when I was working on it, having never used machine learning, I kind of ran into a wall because I like to think of myself as a theorist. So I thought I would be kind of like Johannes Kepler with this huge grave rob data set from Tycho Brahe. And I'd sit down and sort of, sketch this elegant theory that explained all of the data a priori. Or failing at that, I would come up with a structural equation model with a lot of direct and indirect effects, but it would be very hypothesis driven, very deductive. But the more I tried to come up with that, the more I kind of drew a blank, until finally I was frustrated and I decided to just kind of give in and don the dunce cap of empiricism, to sing like Doris Day, que sera, sera, whatever will be, will be. And it's this enormously liberating experience where even if you do have the ability to come up with really great theories and ideas for what's happening in nature, to use some of the virtues of machine learning to see whether you can recreate those patterns without trying to figure things out beforehand. That's my best explanation of it. And so I want to talk about what random forest offers to the perplexed empiricist, the person who doesn't know what's going on. And I'm thinking in terms of three main virtues: performance, simplicity, and flexibility. So performance is pretty obvious. When you got a global data set, you want to predict a large proportion of your data. You want stable models. You want something that's highly predictive. Simplicity, you've got all these potential drivers of your response, in this case, symbiotic state. What you want to do is separate the wheat from the chaff. You want to find out which variables aren't pulling their weight and get rid of them. And finally, there's flexibility. So talk a little bit about linear versus curvilinear models. When you can't specify beforehand what shape you think the response is, it's nice to have something that fits it kind of flexibly, something that has the ability to fit different kinds of nonlinear or curvilinear responses. And so those are all things that I wanted beforehand to see if I could, empirically drive something that made sense about these forests. So I came to random forests, and I did it kind of with dread because I assumed they would be really difficult to implement. And without getting too far into the bolts, I'll say that what I know about random forests, having been explained multiple times how they work, is that it's a consensus model, building lots of decision or regression trees using different features, different predictors of your response variable, and then allowing them to fight via mob rule for majority voting and voting on a final class. So you're generating lots of different models and you're using them to predict, to make a final prediction rather than building one single model that you select. And that sort of concludes the pedagogical part of this lecture. So thank you for listening, but that's about as far in it as I could go because I'm the reasonably bright ecologist who just wants to figure things out. And what I learned is that random forests are very easy to implement for this sort of perplexed newbie. So with a single line of code, I use my data frame with a lot of different environmental predictors and soil chemical predictors. And I just wanted to see whether I could make a model that predicted the data well. So we tested the performance. And I was really happy to see, you can see in this plot, we have the map at the top, which is all the location of the response data that we have. And then the bottom, we have on the X, the predicted proportion of ectomycorrhizal trees and the actual proportion. Is that using a single model, we could predict really well turnover from 0 to 100% ectomycorrhizal trees across different continents. So we were able to recapitulate this pattern of turnover in a lot of different separate places, which are kind of like separate natural experiments. So I was very pleased when I saw this, especially since this took a very short time to implement in R. And the next thing is I just wanted to see, well, what happens if we extrapolated throughout the entire world forest system? And again, this was quite easy because our training data set was sort of vast enough that it was stratified along long gradients of different environmental and soil chemical gradients. We were able to make predictions across huge areas. And so we came up with this map, sort of more or less on the first day of implementing these random forest algorithms. We had a pretty good map, a map that looks a lot like our final one in publication. And then we came to model selection. So in this case, what I wanted to do was sort the importance of all these different potential explanatory variables. And so I made a variable importance plot. And right out of the box, these random forest libraries give you the chance to sort variables according to different importance metrics. And what I could see when I looked at these is that there's a few variables to the left that are very important. They have this high ink node purity. And then there's this long right tail of variables that basically aren't doing any work, or that look like they're not doing any work. They're all of a much lower importance. And we can quantify what would happen if we remove those variables. So here we're looking at the error coefficient of variation on the y-axis and the number of variables. And they're listed from the left to the most important and to the right to the least important. And what we can see is that there's an inflection around 4 variables, meaning that our models reduce the prediction area pretty precipitously up to 4 variables. And then after 4 variables, there's not a whole lot of extra performance that the other variables are offering. So I thought, oh, that's pretty good. Not only do we have a high performing model, but it's most weights being driven by a few variables. Again, this is very early in the process, so able to do this quite quickly. So then I wanted to inspect the model. And here I wasn't sure whether this was something that could be done quickly or whether this was something that was quite difficult to do, because I'd always been told that random forest models were kind of a black box. But I was quite thrilled to see that actually through these partial feature contributions, we can essentially do the same sort of plots we would use in any multiple regression model. We can look at the influence of individual variables in a random forest model. And when we did this, I'm not going to dwell on everything here. What we found is that even though I can't really describe very well how these partial feature contributions are derived, I can interpret the outcome pretty easily. And when I looked at it, I saw that if we look at the proportion of ectomycorrhizal trees, it's high when decomposition rates are low, and then it drops precipitously along this sort of critical transition period. And this is what one of these plots looks like. So high ectomycorrhizal area when decomposition is low, and then it drops. And we can look at this differently. We can look at how temperature on the X and precipitation on the Y affect symbiotic state. And then we can put our predicted proportion of ectomycorrhizal basal area. When we do this, we see the same thing. There's this really large transition that happens. These K values with lines are kind of like isolines. So these are the decomposition coefficients fit to these combinations of temperature and precipitation. And basically, you can see these red dots are when decomposition is really slow. It's cold and dry, so it's slow, and it favors ectomycorrhizas. And then there's this rapid transition. And so I was sort of pleased that using all this information, we could come up with a pretty simple story, which is that there's this really critical rapid transition between ectomycorrhizal forests and mixed forests that contain some ectomycorrhiza but aren't dominated. And it seems to span some of these ecotones between biomes that are dominated by different symbiotic groups. And I'll say in a fairly short time, in a way that is not sort of computationally daunting, was able to come up with this model. And we even named a new biogeographic after Sir David Reed, who's sort of a luminary in the field. Climates that slow decomposition favor tree root symbiosis with advanced decomposition abilities. So that's saying cold and dry climates favor those plants that associate with the ectomycorrhizal fungi because they're good at obtaining nutrients when there's a lot of climatic barrier to nutrient release from decomposing plant material. And I guess in summary, what I want to say is basically that these models were not terribly difficult to implement for a quantitative noob. And I was able in a very short time to come up with a very predictive model using just this sort of empiricist dunce cap without having a lot of a priori expectation. And it's kind of my feeling that if I can do it, anyone can. I've become an evangelist for random forests. And so I think that when we talk about teaching our students and teaching new people how to use this, we kind of need to do so in a more accessible way, and that this ought to be sort of a toolkit that any ecologist can use. And that's it. I'd be happy to take some questions.
Thanks, Brian. That was super interesting. So while we're waiting for questions to come in, I don't see anything in the chat yet, but so I have time to sneak in my question. I maybe missed this, but do you have anything that could be called a plant trait in your model, like plant tissue nitrogen or something like that? And I guess if so, or if not, do you think that kind of thing would help in this kind of situation, distinguishing the different fungal symbioses?
I think it would be quite interesting. We didn't put any plant traits in. The only sort of response trait that we're trying to predict is whether they associate with one or two of these functional groups. There have been others like Colin Averill who have looked at different trait syndromes that are consistent with the ecto versus the arbuscular mycorrhizal states. And they found, for instance, that senescing leaves of ectomycorrhizal trees are lower in nitrogen, and senescing roots and ectomycorrhizal trees are lower in nitrogen, which leads to some interesting hypotheses, I think, when you combine it with our data, which is that it seems almost as if ectomycorrhizal trees both thrive in climates that slow decomposition, and they possibly also have a biological downward control where they promote leaf litter that is resistant to decomposition, leaf litter that is nitrogen poor and so is slower to decompose. And so there might be, you know, sort of this positive biological environmental feedback on the effect of decomposition.
Thanks. Okay, so maybe we can do one quick last question, which is David is curious how you performed your cross-validation.
Yeah, so we did a few different things. There's a k-fold cross-validation where you force your models to become naive to different amounts of the training data, and then you see how well it predicts the full training set. So we did that first with withholding 10% of our response data, training random forest models, and seeing how well they predicted the remaining, all 100%, and they were quite stable. And then we did a more aggressive sort of semi-spatial cross-validation where we took the number of plots we had in Africa, which was our least sampled continent, and then we rarefied all of the continents down to the depth of Africa. And then we saw whether our models would train. And in that case, we were throwing away more than 90% of our data, but we had only something like a 10% reduction in performance. So we were still predicting about 70% of the variability. We tried a few more things. A lot of it is listed. Our supplement in this paper is quite long and really goes into the cross-validation